right. So I just put a title on it. This is today's research meeting. And the topic today, well, I'm going to talk about the hippocampus and hippocampal complex, but we got here because we're talking about learning temporary structure. It turns out that we spend a lot of time in our lives, most of our lives are just looking around and seeing where things are right now. And I'll talk about that. So, these are not necessarily permanent structure in the world, but we constantly learning temporary structure in the world. And so this is basically some stuff we talked about at the London Focus Week, but also stuff we talked about in prior discussions. So, so I'm just going to jump into it. Please interrupt, ask questions. I'm going to start with a little. I thought I'd start with a little neuroscience, neuroanatomy on this, just so everyone knows what we're talking about. This picture here. This is a sagittal slice of the brain, and it shows, you're basically from nose to the back of your head, and the hippocampus is right here, sort of at the top of the brain stem, and it's relatively small compared to, let's say, the cerebellum or the cerebral cortex. If you then took a different slice, the coronal slice, where you're, I think I spelled that wrong, where you now go from ear to ear. So now this is a typical view. I actually don't even know what animal this was from. I just found this online. But it's the same idea for all mammals. What you're looking at here is this is a sheet of tissue. And if you were to go in this sheet of tissue, continue, this is like in the center of the brain here. And this is down here. If you follow this, it becomes the neocortex. And then you got your neocortex going around, top of here like that. So the neocortex, its 6-layer structure comes over here, and then it folds into this, and it follows around and does this wiggle into a little dance like this. This bluish area here is the entorhinal cortex. These 3 green areas here are classic hippocampus. And so these are the things that that we talk about a lot, associated with episodic memory and short-term memory and things like that. And these are also what we believe are sort of analogous to what's going on in the cortical column. So if you look at this picture on the right here, the grid cells we always talk about are in the entorhinal cortex, or in this media part of the entorhinal cortex, which is this side. And, these are like where locations are. There's the lateral entorhinal cortex, which is not as well understood, at least I don't understand as well, but they often talk about like this is like what and when. So this is more like episodic memories, where this is like where things are going. On its own, this might seem like a little bit like a cortical column or a learning module because it's got like what and where. But generally, the idea here is that place cells, which are specifying like features that you can observe in a space, those are in CA1 and CA3, and also in this little thing called the dentate gyrus. And there's this thing in the middle here called the subiculum, which says head direction cells. But this is all very fuzzy. There's so many variations, so many, nothing is clear in the brain. But this is the general rule. And someone had once proposed that that the cortical columns came about by taking, because these things are interconnected. And so someone once suggested, hey, this is sort of like a cortical column. You've got, this could be the lower layers, these could be upper layers, these could be middle layers. And then this might have been a prototype for how later on the cortex became a 6-layer structure. I don't know if that's true, but it's a nice story, and it helps you think about it a lot. So I don't mind thinking of it that way. But it's certainly believable that evolution could have taken a more complex, evolved structure like this, simplified it, made it into one thing, and that became the cortex, the cortical columns in the cortex. So this is the thing we're talking about here. If there's any questions, let's just keep going. I'm not looking for people raising hands, so if you want to speak, you have to just shout out. This is their classic Feldman-Vinessen diagram, where they showed the different regions, and this is the visual regions of the monkey's cortex. And they always show that the entorhinal cortex is here at the top, and then even above that, further above that is the hippocampus. That's the hippocampus, and that's the entorhinal cortex. And so we all bought into this idea that, okay, information comes from the retina down here, goes to the thalamus here, and it goes up this region of all these different processing until you get to the top, and that's where, the top of the pyramid is. We now know this isn't true. These connections do exist like this, but as we've outlined in our hierarchy paper, and other people have documented that all these regions, many of these regions get input from subcortex. Many of these regions get input from the retina. They all have motor output. So these are operating in parallel, not just hierarchically like this. And information doesn't have to flow like this to get all the way to the top. These regions all get activated very quickly, faster than it could take. So this is a misleading diagram. It's not wrong in sense these are the hierarchical connections. So these are establishing in our current understanding when you have compositional structure, like, if this region is projecting to this region and back again, well, these can learn compositional structure locally here. So that's not incorrect. And so that's what they're plotting here. They are showing the hierarchical feedback and feed-forward connections, but they don't show all the non-hierarchical connections which go between all these things at once, so between multiple regions at the same time, horizontally and so on. And they don't show the subcortical connections, so it's misleading. And it took us a long time to sort of shake ourselves of thinking of it that way. Any questions about that before we go on? Okay. Very famous patient. You almost all certainly heard of him. For many years, it's just known as HM, but now after he died, they revealed his real name, Henry Molaison, I guess. He was an unfortunate guy. They removed both of his hippocampus. There's a left side and a right side. And because he was having epileptic seizures, and they had success with stopping epileptic seizures by removing one side of the hippocampus, and he had them on both sides, so they removed both of them. That was a mistake. Because after his surgery, he could not form any new memories. He could remember things about a month before his surgery. He had very good recall of those things, but couldn't remember anything new. I mean, moment to moment, second to second. And so he was studied a lot. And this was a genesis for a lot of research that came along that where people say, well, the hippocampus is where new memories form because he couldn't form new memories. His old memories were there. So the cortex is the locus of more permanent memories. We know that the cortex learns slowly, and the hippocampus learns very quickly because these new memories form very fast. We also know that the cortex requires the hippocampus to learn. That is, without the hippocampus, you can't even form long-term memories. Now, there's some exceptions that HM actually was able to learn some things. He just didn't know he learned them. And he was surprised when he said, I didn't know how to do that. How did I learn that? So this is not quite 100% right. There's also this concept of episodic memories, which when you have memories associated with particular times and places. So if you talked about this before, if I'm learning something new at first, like, oh, here's a new coffee cup. Initially, I'll remember not just the new coffee cup, but where I was when I learned it and what time of day it was, the environment in which it occurred. Those episodic memories go away. But even though the memory of the new coffee cup will stick around. So episodic memories are in the hippocampus. So these are some of the things that people have observed over the years. And one question I've always asked is, well, how does the hippocampus learn so rapidly? Because you can form memories almost instantaneously. One exposure, in a matter of seconds, you can learn things. Where did I just put my keys? Or I just left them on the other table 2 seconds ago. So that always was puzzling because synapses where memory occurs, and we now know that memory occurs when you form new synapses. They can't form quick enough. They can form very quickly. There's examples of synapses forming in the cortex in a matter of a minute or 2 minutes, but that's not fast enough. So we speculated that I speculated that for the hippocampus to learn so quickly, the hippocampal complex, that there must be existing synapses that are silent, that aren't doing anything. And this learning, it's just converting an existing synapse that isn't doing anything to one that actually does something. That was very speculative when I first proposed that. I came up with that. But it's pretty much been verified, I think. These silent synapses are real things. They exist in great numbers in the hippocampus. They exist elsewhere too. I was just talking to Subutai yesterday. They find them in the cortex too, but mostly when you're young, and they kind of go away. But I'm going to read more about that. So one reason you can argue this is that in biology, having these sort of silent synapses is expensive. They're taking up space, lots of space and lots of energy, but they're not doing anything. Whereas the synapses in the cortex are there because they're working and they're doing something. So you could say that silent images are expensive, and maybe they're split up this way, that they would put all this fast memory in the hippocampal complex because you can't do it everywhere because it's too expensive. And so maybe this division between quick learning and slow learning is a biological necessity, but it's not clear if that is a distinction that's needed for Monty. We don't have that constraint. We don't have to pay a penalty for, changing a 0 to a 1 in some memory structure. So that's just something to keep in mind. Everything I'm going to talk about now is biology, and it may not be applying to Monty. We may not have to do it this way. This is how biology does it. So we're going to start with that as our basis. Before I go on, any questions about that? Maybe this is all review. I don't know. Okay, let's talk about grid cells. In particular, grid cells, of course, we propose that this is what's going on in the cortex in each cortical column, and there's a lot of evidence for that, so it's pretty certain that this is right. But grid cells were discovered in the entorhinal cortex, and just a couple of things about them. They're in these grid cell modules, and there's a series of grid cell modules that, as you move along the surface of the entorhinal cortex, the medial entorhinal cortex, you see a grid cell module, then another grid cell module, another grid cell module, another grid cell module. And each successive grid cell module has a different scale. So they get these little dots represent where a particular grid cell, one cell, would respond in, say, an environment, in multiple occasions in an environment. And then another grid cell module, a cell would respond to multiple places, but they're further apart. Another one, they're further apart still, and so on. And so you have a series of these aligned in a row. This is the picture we've looked at many times. It's one of my favorite images on the right here. And this is showing from experimental data what one of these grid cell modules looks like. So this pink outline, and the blue outline's another one, and the blue outline's another one. And I spoke to the scientist who did this tank, and he confirmed that these are different scales. So just like you see here, and we can go into the details here. We've discussed this before, but within a grid cell module, there's multiple cells that are active. So there's 6 little fields here. So instead of one cell you have like maybe 6 bumps of activity that are moving around, but they achieve the same result as this. Now, this is something I really kind of find funny. Tank, the scientist, told me there's only one grid cell module per scale. And these are pretty small. These are like cortical column size, or small cortical column size. You can see the 120 microns here. So this might be like, I don't know, 400 microns or 500 microns by 250 or something like that, 300. So these are kind of like small cortical columns, but it's really questionable. Like, there's only one per scale, because that just seems really small. Even though the entorhinal cortex is not very big, it seems pretty small to me. But that's what he said. And so I've been working with that. And I'm just thinking about preparing for this presentation. Like, God, is that really true? Can it be that small? But that's how I've been thinking about it. You got one module for scale, and they're arranged in a row. And this could work okay, but we're asking these modules to have a lot of memory, to be able to learn a lot of things, just like we ask a cortical column to be able to learn a lot of things. So it's always something that's a little bit slightly worrisome to me, but at the moment I don't see it as a fundamental block, but maybe something we'll have to deal with later. Okay, so that's the end of the empirical data. Again, any questions about that before we go on?
Well, just a quick question about the silent synapses. These still have to be learned by association or somehow. I get the point that like if they're silent and the hippocampus is able to turn them on very quickly, then we'd be able to form instantaneous memories. But we still have to form.
They're formed. It's like they're formed randomly. So, and there's lots of them. So let's say a typical cortical pyramidal cell might have, let's say, 5,000 synapses on it. A typical hippocampal cell might have 30,000. So there's a lot more. And the idea there is, in normal learning, you could form a synapse between 2 neurons by growing it, moving them together and growing the synapse between them. In this case, it's sort of like there's random connectivity. It's not learned. It's just overly connected, and you're selecting the ones that are useful. That's the basic theory. And I think it can work, it will work, even though there's some intuition about like, hey, is there enough of those connections and are they sufficient? And maybe it's denser representations and things like that. But I don't think there's any other way around it. And that's pretty much, so I don't know if I answered your question, Rami,
Yeah,
but
yeah. And this is the part
uh,
that I was missing. I assumed that all synapses would be learned using the same
no,
Hebbian.
right. No, it gets just, yeah, you have two populations of cells and you can learn connections between them by having the axons and dendrites go closer to each other and then build this little connection, the synapse. And this is just saying, oh, we're just going to overly connect everything. It's very dense. I was talking to Subhatai Ahmad yesterday, and he sent me a couple of papers. I just started to review a lot of new research about silent synapses and how many there are and where they see them and so on. Um, and one of the things that comes out is that when new neurons form, wherever, they tend to have lots of these silent synapses. So when you're in your development periods right after birth, all the synapses, even in the cortex, have lots of these silent synapses, and they get paired down and selected. They also grow and form new ones. And then throughout life in the hippocampus, one of its things is that it's always creating new neurons, new pyramidal cells, which were the first time they really discovered, neurogenesis, meaning that you constantly form new neurons in your life, is in the hippocampus. So again, here you can imagine the hippocampus. It's basically saying, I'm always going to be learning something new. So I'm always going to be putting out new neurons, and those new neurons will have lots of silent synapses. I'll figure out which ones are good. And then over time, those neurons will go away. I'll get some more neurons, and they'll have lots of, so we're constantly looking at new possibilities of connections, and it looks like it's yeah, I think I've said enough. It looks like when any new neuron comes along, it has lots and lots of these silent synapses, and then they either are useful or not, and then they get
Yeah,
pared down. um,
makes sense. Thank you.
yeah, okay. So these next things here are some more introspective observations. We've talked about these in research meetings. It's just I don't know if they're not controversial necessarily. They're just things that just if you think about them a bit, you go, oh yeah, I got to think about that. That's pretty important. So it seems to me learning anything requires attention and forms episodic memory. So this is like I try to come up with an example where I learned anything new, where I didn't have to attend to it in some way, and I didn't have any kind of episodic memory for it. So even the simplest little things, if I'm going to learn a new, if I'm going to learn a new flower or a new pen or a new anything, I have to attend to it, and I'll have an episodic memory for it for some time. Oh yeah, that was 2 days ago. We ran across that, new fork or whatever it was. And this is also reinforces that new learning must occur in the hippocampus, the hippocampal complex. That's again, HCC stands for the whole thing: entorhinal cortex, cerebellum, and hippocampus. Um, and so that's consistent with what they saw with patient HM. And then I've talked a lot about this in the coffee shop, looking at intersections, but we are constantly learning new compositional structure. I've come to realize this is 99% of what we do in our life. We're just constantly building, observing things, attending to one thing after another, and learning temporary structures of where things are. Because often so much in the world is changing all the time. And we tend to focus on the new things, and then we're just constantly saying, "Here's something new. Here's something new. Where is it? Where is it? Where is it relative to something else?" So this is compositional learning. That's all it is. We're learning where some object that I recognize is relative to some other object. Where's this new car in the intersection? Where are the people in the coffee shop? Where are the dishes on the table? Where did I leave my keys? That is a compositional model. It's like my keys were on that table in the living room a moment ago, and they're not going to be there again, but that's where they were a moment ago. So this is really what our lives are about in Monty in the future. If it's going to be an autonomous being, robots and whatever doing stuff, they're going to have to do something just like this. So this told me for the first time, I realized after thinking about this, that the column equivalents in the hippocampus must be learning compositional structure, just like the cortex. It never occurred to me until we understood compositional structure that that's what's going on between those regions, especially like the different grid cell modules and the different associated hippocampus place cells, that is also learning compositional structure. And that's, where's the keys? Where's this kind of stuff? And so that was a big sort of insight to me, just to get a deeper understanding of that. And then this is just, I think, repetitive, perhaps. The cortex. No, this is not. This is different. Like, but when we learn new compositional structure in the hippocampus, like where the cars are in an intersection, so on, it requires the cortex to recognize the objects. So the cortex says that's a car. The cortex says that's a person. The cortex says that's a bicycle. But I can't ask the hippocampus to do that. The hippocampus is not getting input from the eyes and the ears and the skin. So the cortex is recognizing the objects. But the hippocampus initially has to figure out what their arrangement is to each other. So there has to be this constant play between cortex recognizing known things and the hippocampal complex showing how they're arranged uniquely at this point in time. So that really sort of set up this interplay between the cortex and the hippocampus, the hippocampal complex, which is shown in this diagram right here. So this last statement here implies this. This is the cortical region in the cortex, which would be recognizing an object. It could be visual, touch. It doesn't really matter. It could be a bunch of stuff. And there's another one, and we know there's a hierarchical composition structure going on here. But somehow, whatever I recognize here, some object, I have to tell an equivalent column over in the hippocampus, or hippocampal complex. Again, this is place cells plus grid cells. I have to tell it. And this says that I had to have these connections between cortex, all these different regions of the cortex, and all these different columns, the column equivalent in the hippocampal complex. And so these green arrows were required based on this observation. So that was a really interesting prediction because I wasn't aware if those existing connections existed or not. Does V1 project to the hippocampal complex? Does V2, et cetera? And so I did some quick search, and yes, they do. I didn't do an exhaustive search, but I found lots of evidence of like, oh, look, we found these connections from V2 to equivalent columns in hippocampal complex. These would be mostly object IDs. We just have to say, I found a car, and this is where there's an intersection. Here's your keys, and here's where it is relative to the table, that kind of stuff. Here's the paper I just found yesterday, which I thought was good. I'm going to read it to you. I didn't read the whole paper yet, but there's an interesting little thing. I'm just going to read it right down here, starting here. I'm going to say, here we show that like hippocampal place cells, many neurons in the primary visual cortex, V1, are freely moving rats. Okay, it's rats, but still rats. They fire at specific locations as animals run repeatedly on the track. The V1 location-specific activity leads to hippocampal place cells activity, both spatially and temporally. The precise activities of the individual V1 neurons fluctuate every time the animal travels through the track, just like place cells, and in a correlated fashion with those of the hippocampal place cells firing at overlapping locations. So they're basically saying we're finding place cells in the V1 that match the place cells in the hippocampus. This suggests the existence of cortical neurons that are functionally coupled to the hippocampal place cells for spatial processing during natural behavior. These visual neurons may also participate in the formation and storage of hippocampal-dependent memories. So you essentially find that these two V1 and equivalent, column in the hippocampal complex are essentially occurring together. They're both mapping the same type of thing. And this is happening very rapidly. So this is not an evidence of direct connection. This is evidence of behavior. But I did find papers that talked about neurons that project from V1 and V2 to hippocampal complex. So this idea here that all these columns in the cortex are directly influencing the equivalent columns in the hippocampal complex, just seems to be a fair amount of evidence for it, and logically it had to happen. If these,
just out of curiosity,
you go ahead.
Did they look at which fires first, the columns in V1 or the ones in hippocampus?
Right, and this one here, it even says it. This one says here the V1 location-specific activity leads
to
lead hippocampal place cell activity. So,
which kind of makes sense.
Right, that's what we'd hope. Right. Well, first of all, I don't know of any evidence that the hippocampus is getting direct sensory input. But it would make sense that this activity here would very quickly lead to activity here, which is what it is. It's very quick.
Yeah, that's nice.
Yeah, and then when you were looking at the connection papers, was there any pattern in like what layers were projecting or?
I didn't, I didn't get beyond
Okay,
the
yeah, no, I was
abstract.
just curious.
I didn't read anything in depth. I was doing like a sanity check, right? A sanity check says I should be able to find some evidence that these things are tied together. If I can't, I'm going to keep looking. But
Yeah,
a
and I
sanity
kind
check.
of yeah, at least at higher levels by definition, I think you kind of have to. Um,
I didn't
well,
know
or like
that.
under most theories of the hippocampus, like if it's going to invoke memories by invoking cortex, like unless the
I
hippocampus was totally essentially segregated from cortex, there must be some kind of connectivity.
for years and years and years, I talked to neuroscientists about this, and everyone assumed that connectivity was right here. You had to go up the hierarchy
Right.
to get there. And I said it too. Right? So everyone agrees it's connected. The
Yeah.
part that was novel to me was that's not the connection that we're really interested in right now. It's these other ones. And
Yeah,
to
like
me, this
V1
was,
and V2.
I could be out of touch. I'm old. I don't remember. I haven't keep track of everything, but I didn't know that these things existed. I had no knowledge of any kind of connectivity like this. So this theoretical speculation, well, not speculation, but this introspective observation that I just talked about here led to me say that, well, these must exist. And so let's just do a little sanity check. And now I can see, a whole bunch of papers, and I didn't read them. I didn't get there yet. But
Did you see, did this paper talk about whether the V1 projections goes to areas with like grid cells with a small spacing or?
I didn't even get to that. I don't know if I didn't really. I read a bunch of abstracts,
Yeah, okay.
and the abstract said, okay, there's connections between
Yeah,
this and this and this. I'm sure I'm going to speculate that it's like most typical neuroscience papers. When you get into it, the details are far more hard to interpret than their summary. And so I don't know yet. I haven't looked at it. What is the rat? What's it doing? Or what was the techniques they used? This one sounds. I have really no idea. This was not a anatomy paper. This was like a physiology paper. Sounds like co-fluctuation and activity. It's worth looking at. But the point is, I felt pretty comfortable about these introspective observations. I felt like, well, they have to be right. It seems like I can't imagine them not being right. They've led to this basically. This has to be true if these are correct. And is there any evidence for it? Yeah, there's plenty of evidence. So now we could dig in deeper if we want and figure out specific details. I could imagine these are more like object IDs because these areas here could be very modality specific, and the hippocampal complex is going to be less modality specific, and so the details here really don't matter. Really, we want to know what are the objects here. So there's more like an object ID to object ID voting. We'll come back to that in a second.
yeah,
Um,
that makes sense
that's
with them activating the place cells and not the grid cells, right?
right. I guess it's. I haven't thought through it deeply yet. So, but to me, this was a total rethinking of the relationship between cortex and hippocampal complex. It never occurred to me to think of it this way. The other thing which is kind of just keep in mind is that in our theory, the thousand brains theory, let's say this is some visual region here. These columns all basically contain the same models, at least some subset of them do. And so there's many columns here that are sort of replicas. That they're individually learned, but they're copies of each other. And so and then why do you have them? Just remind, the reason we have all these columns that have the same models is so you can do quick inference. That's the whole point of these big regions. It's quick inference, so you don't have to move. A single column here could recognize something, just like the ultrasound probe we were talking about, but it has to move around a lot. And so the reason you have all these multiple columns, and so you can do flash inference. And that's pretty much the reason they have so many columns in each region. But once you've passed inference, once you've inferred something, you no longer have to have lots of columns per region. You could just have one column per region because that's all you need, one learning module. So this is where I'm starting wondering, is there enough memory here for these things? And, so this idea that you could take, thousands of columns to recognize something quickly, but now you can invoke the same model, a similar model over here very rapidly. in one column, it kind of makes sense, even though it sounds hard to believe a little bit.
Yeah, and I guess it
Uh,
helps maybe with the kind of transfer, or not transfer, but like, yeah, learning first happening in the hippocampus. You could imagine maybe those representations eventually disappearing as the cortex kind of takes them up.
right. The cortex,
And so
right.
it's almost like wiping the slate. But I mean, you don't remove everything, but like, I don't know, like, yeah, the first time you see like a particular mug, you're kind of in V1, you're recognizing local kind of features and stuff like that, and building like a pretty complex model in Hippocampus at the same time. Um,
Right.
but
It's
then
like it's
eventually, like a column will recognize that, and then there's no need. All the Hippocampus at that point cares about is the ID of that weird mug or whatever.
yeah, I didn't follow all that. I thought you were going in one direction, and maybe you went in a different direction. I thought you were going to say was let's say this guy recognizes the mug and this guy recognizes the logo, and the first time we see them, we start, we see, oh, there's a mug and there's a logo. These connections can be slow to learn. And so just because you saw it here, you don't have enough time to form these connections reliably, but over here you can. And so this says,
Yeah,
oh yeah, I'm at the mug, I'm at the logo. I can form this connection instantly. I may have to see this maybe 20 times over here over a couple of days before I really establish these hierarchical connections. So this fits with the idea that I think you use this term too, Niels, about the scratch pad, scratch pad. You're right here, consistent with the scratch pad hypothesis. That we're in a little temporarily learning where scratch pad, like, oh, where was this relative to this, and then this over here slowly develops those learnings later because they take more time.
yeah,
No,
yeah. So
I don't
I
know if
think yeah, Vivian
was that you were talking about? Were you talking about something else?
Kind of. So yeah, I think Vivian's used that term. But the what I was talking about was that yeah, so you need the hippocampus to learn it, but then let's say through replay, maybe while you're dreaming or while you're awake, or yeah, seeing the mug several times, slowly cortex learns it. At that point, you don't necessarily need the model, or at least such a rich model in
Right,
hippocampus. And so maybe that helps it kind of flush these older
right,
representations to
right,
then learn new stuff.
right.
Um, and then
I got a
yeah,
little image and
it,
discussion
it, it
about
was
that.
more just this question about like capacity that,
Right,
okay, we
right,
have thousands of columns on the left, but what do we do with the hippocampus? I guess to a certain degree, kind of flushing representations once they're learned in cortex could help.
right. So we're going to talk about. We talked about this in the offsite. I'm going to talk about it again a little bit more in a second here. Um, but the basic question is this a scratch pad where you're constantly throwing away the piece of paper and taking a new one out and starting afresh? And this is the printed book, or do these do I have a permanent memory of coffee cups with logos on them over here? I've learned it here. Does it stay here forever, or do I throw it away and but keep the permanent ones over here? Um, that's related to I think the question you're just asking,
Yeah.
and that relates to capacity because if I don't have to, I don't have to store everything over here. In fact, the evidence we would have is there's far more cortical regions than there are equivalent columns in the hippocampal complex. There's dozens and dozens and dozens over here, but there's, 10 or 12 or 15 scaled columns here. So there's many more of these, and so it's not necessarily just one to one like this, which I'll come to in a second. As we talked about this in London a bit, and also as we talked a moment ago, you're always learning new cells in the hippocampus, and so in theory, if you're learning new cells all the time, some of them are disappearing because the hippocampus doesn't grow throughout life; it actually shrinks. Um, but so in theory, you'd be forgetting things over here all the time. So this is really a scratch pad memory. It works for a period of time, and then it goes away. And then you have to rely on anything that's really worth learning permanently or more permanently happening over here. Okay, so now we're getting into questions. This is exactly the question you're just asking, or we were just talking about, and you asked about. So here I say columns store previously learned objects, and do the column equivalents, these little boxes in the hippocampal complex, do they also store previously learned objects, or they just store novel and temporary variations? So that was the exact question we were just talking about. Is this really just a scratch pad where you forget things, or is this a place that's fast learning and you can remember things here too? So the evidence would suggest it's a scratch pad. The evidence would suggest that just you're going to throw things away here. I gave you the example. There's many more columns than there are. There's many more regions than there are columns in the hippocampal complex. There are new cells being formed here all the time. These, and therefore things must be forgotten. There's very little neural tissue here compared to over here. So those are the evidences we have for that. So this puzzled us when we were in London. Like, okay, well, if I recognize a logo here, and I want to invoke a logo model here, but there is no logo model here because this is a scratch pad. I no longer have a model of a logo here. Maybe I had it once when I learned a logo, but it's gone. It's now over here. So how do I see? I have a mug, I have a logo, but I don't have models for mug and logo over here. How could this work? How do I invoke, if this is a scratch pad, I don't have all these models over here. If I did have all the models over here, it would be easy. I would just do our typical association that we do for voting, like take a model here and a model here, and you associate the IDs between them. But I can't do that if this is really a scratch pad memory. I can't assume that the models I have over here match the models I have over here. So we puzzled about this in London, and we came up with an idea, which I'm not sure is right or not, but it's kind of an interesting idea, and that's represented here. I said, could unique object ID determine grid cell anchoring in the hippocampal complex in a model-free way, similar to how a seed can determine a random number sequence? So the idea was, imagine you have this SDR, which is an object ID, right? An SDR, it's an object ID. In the cortex, if you know the object ID, you also know how to anchor the grid cells. And once you anchor the grid cells, essentially you've defined the object. That is the definition of an object. Everything then is relative to that object reference frame. So now imagine if I took that object ID and I send it over here, and I don't have a model here. I've never learned the association between a model here and a model here. It's not an associative learning. But what if this object ID on its own could, acting like, it could determine what a unique reference frame anchoring, meaning the grid cells would anchor based on, if I give you that SDR, I uniquely find the anchoring, in which case I don't need to have a model of the object here. The ID itself establishes the model. It doesn't have the data. It doesn't have what features, but it says, I know the reference frame for this unique object. I don't know what the features of that object, but I know, I've established a model and I can start adding features to that model here. And I'm going to guarantee every time I see the SDR, which is the object ID, I'm going to establish the same set of grid cell anchoring here. And so you start off with a blank model, but it's equivalent to these models. So as long as I have the object ID, I'm going to guarantee to have the blank model equivalent. And now I can start adding features to it, which is what the whole point of the hippocampus is to learn these temporary arrangement of features. Like I can say, oh, okay, well, I've established, so imagine this is the mug and this is the logo. I don't have models for over here, but when I send the mug ID over, I establish the grid cell reference frame that will be unique to a mug. I establish a grid cell reference frame that will be unique to a logo. And now I can start saying, oh, well, look, there's a relationship between these two. I'm going to learn that rapidly. So all I've learned is that over here, I don't have a full model of the mug or the full model of the logo. I just say, well, I do know that in the reference frame of the mug, there's a logo ID at that location. That kind of thing. I know it's a lot to absorb if you're not thinking along these lines all the time. But maybe I should stop there and let people ask questions for a bit. You can say, I'm totally confused too.
I don't think it's the same thing as anchoring, but Fieta Lab from MIT, they have a computational structure where it can accept any sensory input, so an SDR in this case, and that input maps uniquely to a grid cell kind of location, which you could interpret as anchoring. It's a place cell, grid cell kind of scaffold that they created, and so you get a sensory input. It activates place cells, which activates grid cells, and they mutually stabilize each other. which kind of
Right.
has the same mechanism of like, as long as the SDR is unique, you end up in a different place in the grid cells. And the
Right.
same one all the time, and it's error correcting as well. So if
I
you're
think
so maybe
right.
that is a similar mechanism.
That is it. It all depends. You have to look at the paper. If they're talking about true SDRs, then it's exactly what we're talking about. If they're talking about just generic features and generic grid cells, it's related but not the same. But I think it's a good observation. I mean, we have this in our current models, Tristan. If I could uniquely identify the the feature ID in some sense, it would uniquely identify the location, and for the object as well. At least this is how it works in our biological model for orbital columns. Again, Monty works differently. Um, but it's related. It's very similar. I would agree with that. It's very similar. But this is going one step further. This is not a feature. Well, it's an object, which is going to be, this is an object, which is going to be a feature on this object, but it's an entire object. And you're saying just here's an object SDR, but it's the same thing, Tristan. I think it's a good observation. It's very similar.
Yeah, maybe just to add a bit of context to this question, I think the reason we started talking about this topic and you came up with this proposal, Jeff, was because we were thinking about, okay, how can we recognize that the set dinner table is set wrong and the fork is in the wrong place, and then specify that we want to move the fork in a different place. And this mechanism would basically allow us to build a quick temporary model of how like the plate, spoon, fork are arranged on the table without having to kind of relearn everything. We can just kind of instantiate the reference frame of the table and then lay down the features individually, temporarily in that reference frame in the hippocampus. And then, learn those variations. And similarly with the chip mug, if you can already have a model of a mug and then suddenly you chip the mug and you don't have to learn a new model of the chip mug right away. You can use the model of the mug in Cortex could invoke a reference frame in Hippocampus that's initially empty and then just stores the chip at a location because that's kind of the new feature there.
Right. I don't know if we worked out all the details in that, but that was great. That was like,
Yeah.
because it basically says you can have your cake and eat it too. You can have your existing models, and you can have variations on them, and you don't have to mess with your existing models right away, right?
Right.
That can be learned much more slowly.
Yeah, and I think one open problem we still had was what if you're quickly learning several variations of the same object? Like, let's say we go to a mug painting workshop and we all get the TBP mug, and then we paint something individually on our mugs. And we can quickly learn these variations. But it wouldn't work if we can only initialize one temporary reference frame for each mug.
Interesting. Yeah.
Yeah, I don't
So
know.
I'd have to have episodic memories of multiple mugs with different things on them, you're saying.
Yeah,
so
I
like
think
maybe use like
during the week,
hierarchy at the I don't know, if you have a sense of where each of these unique mugs is in the room, that might partly help because that one can reinvoke the appropriate one.
It's
But,
interesting.
yeah, they still need to be
I
unique.
wonder how many unique things you can memorize. I can memorize a lot of unique things temporarily memorize. Um, you know, let's go back to a different example. Like, okay, where did I leave my keys? Right, perennial problem. Right, so that's a temporary compositional model. And as soon as I can't remember where I left my keys, yesterday. But as soon as I can't remember where I left them 2 days ago, 3 days ago, 4 days ago. I mean, unless I haven't moved them in since. If I haven't moved them in 3 days, I might remember where I left them 3 days ago. But if I moved them yesterday, I forgot where I left them 3 days ago. So there seems to be a limited buffer of how many times in the past I can remember something, the episodic side of it. And there seems to be something similar might be with the chip mug. Like, oh, here's the Memento mug or Thousand Brains mug, and it's got a chip. Okay, that's interesting. Here's another one with a different chip. Oh, that's interesting. Now I got two. How many of those could I remember? Or how many unique quick variations could I keep in mind at once without learning permanent models of them? I don't know. They might be limited.
Yeah. Yeah, and I
They
think
might.
what you said, Niels, about maybe their context in a scene or something could help too, because I think the example we talked about during that week was having two identical coffee tables in the room, and the two tables have different objects arranged on them. And so maybe like one of them is my couch table and the other one is the, I don't know, side table or whatever. And
Mm,
Yeah, and you
maybe
can
in my mind, they
imagine
still have
back to
slightly different representations based on where
right.
they
but
are.
it,
You can
right.
imagine if the seed is like a combination of the object ID and its location, I don't know,
Mm.
an egocentric space or something like, by definition, it will result in a slightly different
I don't remember talking about that. I'm sure we did. I just don't remember. But it's an interesting question. It's like, yeah, I have these two little tables in the room. Maybe there's side tables inside of a bed or something. I don't know. And they're identical, so I have a model for each one. But I know them uniquely. When I see one, I know it's different than the other one, and it's in the context of the room. But then I might have different features on each of those tables. So it's like I don't
But yeah,
know.
if it was freely floating in space without that like environmental context or unique kind of child objects, then yeah, you'd have no way of distinguishing them.
But yeah, I think
Right,
bedside tables are a good example because they usually look exactly the same, but they usually have different objects on them depending on which side.
right.
But you think of them
And
as two distinct things. This is this person's bedside table, and this is
the other,
object.
and Jen, Jen has tissues or whatever, and I have, whatever, my
Yeah.
phone, something like that. And yeah, and so when you look at it, it's funny because when you look at the table, it's not like, oh, this is that table. This is a particular version of that table here. Right? They may be identical, but this is the one on this side of they're somehow unique at the same time. Um, we don't have any trouble with that in the context of the room. We say, oh yeah, well, I can have two of the same features on an object, and so I have these two table features in the object. But if I'm just attending to the table, I wouldn't know that. I would just say it's the table. I think I don't think I think that's an interesting one to think through a bit more. It doesn't seem fundamentally a problem. It seems like a tweak, a variation on this issue.
Yeah.
Um, but
Um,
And
not
yeah,
like,
but
oh,
then
Also,
I guess in terms of, oh yeah, it would just say in terms of like forking, just to highlight, yeah, that I guess the way we were thinking about this when we first talked about it was, yeah, with the chip on the mug, you would really attend to like the new stuff, like the chip itself, or maybe that there's a different logo or whatever, and that's what's going to be learned over kind of rapidly in the kind of hippocampal area. But it would avoid trying to like relearn, or just naturally, it would avoid kind of relearning the entire mug. Um,
right.
and
We
and yeah,
didn't.
and obviously you can still do inference and recognize, oh, this is a mug. But it just because sometimes we've talked about, okay, with now you see the chip mug, is that like a new state in the column that kind of conditions everything? Maybe that could happen in the long term, but at least in the short
Right.
term, you don't need to
Right.
rely on
I
that.
think that's a really good point. We basically can take variations of existing objects and store them here temporarily without messing up the original objects. We still, though, eventually, I'm going to learn. I really do remember. I have a really chipped, 8,000 brains mug, and it happened naturally. I didn't do it, but I have it. It's upstairs, and so that's now over here someplace. I don't remember when I first saw it, but I now have. I know that mug. It's different than the others. We haven't explained yet exactly how it is. I have two of these representations of the generic mug and the chip mug over here. All we did say is that temporarily, while I'm seeing something novel, I can learn it here without messing up over here. But I have time how do I clone the mug with the chip on it over here somehow? I don't think we figured that out yet, did we? I don't think so. But this is definitely a step in the right direction. This is like, okay, this is a key ingredient to how we form models of variations. We start with as a temporary scratch pad without affecting anything. And now we have the luxury of time to relearn stuff over here as we need to. So
Sorry,
this is
Jose,
not
you
a
were gonna.
yeah, Jose.
I was just want to just make sure that I'm understanding. So we're proposing that the object ID from the cortical column is supplying a unique root cell anchoring to the hippocampal complex. And that's supported by the paper that you posted in the above, where the V1 place cells activities correlated with the activities in the hippocampus.
Well, it's just be clear. This was a theoretically derived prediction.
Right.
This was not based on empirical observation. This was based on
but I mean,
theory and introspection. I went to the logic here. I talked about these things. These things, and if they're true, and if I can't figure out why they're not true, it leads to this need for somehow the cortex has to invoke the model here. And it also, if I assume that these columns here do not contain all the models, then I have to have this signal itself has to somehow uniquely establish a reference frame here without any other evidence that's always going to, every time I see the mug, I'm going to get the same reference frame, even though there's no data over here. It's going to establish a blank mug model. So that's right. And then this piece of evidence here was this thing here was really just to argue that there are connections between cortical columns, including low-level cortical columns to the cortex. I don't want to read too much more into it beyond that, but clearly, like these things are working in lockstep together. So when this is something, this is and that was this little reference was talking about place cells, and that's not what I'm talking about. I'm talking about IDs.
Right, SCRs.
So maybe I don't know.
There's a thin connection, basically, but
I don't know.
Yeah.
It's more. It's gonna. Like in so much in neuroscience, you have to spend a lot of time looking at the papers to figure out what the hell they're talking about. But on first blush, it says, these things are connected together in a way that I would think many neuroscientists wouldn't have expected. So we got there theoretically; they got there through empirical observation. So it's somehow nice.
Yeah. So maybe this is. I guess, assume, so what's the implication of this? So once the hippocampus gets a root cell anchoring and it can pick up, so I guess, what's the next, I guess? Yeah.
Well, I think we just heard the one big idea, which was that as we think about in Monty, we're going to have systems that learn rapidly and have these same attributes, where they're learning rapidly, but we don't want to form permanent models. Imagine a robot who's given a task to build something. It's going to have to remember, where did I leave the tool? Where's this thing? What was that thing? What did I just do a moment ago? That kind of stuff. So it's going to need this sort of rapid memory that is not permanent. Or at least doesn't need to be permanent, because it's not useful. After a minute later, it's not important anymore. So we need to have some kind of structure. What this tells us is that, at least in the brain, the temporary arrangements of things are handled separately than the permanent memory of things. So we're not trying to, you could add, for example, some temporary knowledge to each of these columns here and have cells that are temporary cells and they forget and things like that. I don't know. It might be hard to do, but you could try to take this functionality and put it inside each learning module. But if that turns out to be difficult, nature said they didn't do it that way. Nature said, I have my own little scratch pad over here, and
I see.
And so it, again, do we have to do it this way? I don't know, but we're going to have the equivalent functionality. There's going to have to be ways that we can learn temporary structures very rapidly, not forget, rely on our existing models to learn those temporary structures without messing up the original models.
I see.
I don't know if it would be helpful to kind of talk through like an alternative model that we also discussed. Which I think you suggested, Vivian, which was also interesting, about having kind of like, and maybe complementary, but like kind of having a bank of object IDs. Because part of the question that we were basically exploring was like, so in this setup, like all the models exist in Cortex. We want to rapidly learn new models in Hippocampus. As Jeff was saying, we probably can't have like just stored all those models. So somehow those models are kind of coming from Cortex. And so, one idea was this kind of seed this seed thing. And I think one intuition for that might be helpful was like, I think you gave the example, Jeff, that like when we had all the objects arranged on a table, it's a temporary arrangement, but you really understand, oh, it's an arrangement of objects on a table. You don't just represent it as like an arrangement of objects in an arbitrary reference frame. Like it feels like it's grounded in this other reference frame.
It's compositional, basically. Right?
It's compositional, but like I guess there's like a structure to the reference frame that it's being ground in, which maybe if you didn't have this seeding, if let's just say you just initialize a random reference frame, and then then you maybe kind of miss that.
Well,
But
it wouldn't work because it has to be, it can't be random, because imagine this is the table here, and these are the items on the table, right? I go in the next room and I come back, I look at the table again. I need to remember the temporary arrangement of the objects.
right.
So this table has to invoke the exact same stuff over here. I can't, if it was a one-time random assignment, then I'd come over here and I wouldn't invoke the same reference frame, and I wouldn't know what the arrangement of objects.
Well, I mean, you could have a higher level hippocampal reference frame that invokes the table when you go into like based on either episodic memory or where you are in space.
But
I think the issue with that is in terms of having the IDs available so that you even know that it's a table as opposed to.
I didn't follow that one.
I don't know if. Yeah, maybe
I didn't follow that. It seems we have these relationships, these hierarchical or these compositional relationships, are tied to the unique IDs here and here, or the unique reference frames here and here. So when I come back into the room, I have to invoke the same unique reference frame here that I had when I learned the temporary arrangement. So I don't know how you do that. I didn't follow it with your argument, but
it was kind of related to the like two side tables that they're in different positions in your room. So if in the reference frame for your room, that would be associated with two different child reference frames depending on which side table you're looking at.
Okay, but
that's in Hippocampus, but that could be in Hippocampus, but that doesn't carry any semantic information about what those objects are.
I think that's an orthogonal problem. I mean, I have to learn those representations of the two side tables over here because they're permanently in the house. I know them permanently. Yeah. It's all here. So to me, I don't see the connection. I might just not be understanding what you're saying. I'm not understanding. It might be my fault. I'm saying.
Maybe I think maybe you're talking about the same thing. I also don't completely recall anymore what we talked about 2 weeks or 3 weeks ago. And the picture I just put there was the only thing I think we had on the whiteboard for the other idea. So not too much to go off of. But,
that's right. I asked maybe
I think the whole point was that if we learn very temporary arrangements, there isn't enough time to actually learn feed-forward and feedback connections between Cortex and Hippocampus. So the kind of green line forward and a feedback connection back. We wouldn't have enough time to learn that. And so that's why it's useful if just given the object ID can invoke a unique reference frame. And then I think the alternative, but actually thinking about it now, almost identical idea was that we did actually learn feed forward and feedback connect, or voting type connections between object IDs. But then the features at location part doesn't have any connections back to the cortex, and this was what is invoked by the object ID and can be learned temporarily in the hippocampus. We wouldn't learn a feedback connections from a specific location in hippocampus back to cortex, but just you can just invoke the object ID, and then that can invoke the already temporary learned model here, or you can learn new temporary things there.
Right. Yeah, and that's kind of more realistic because then you can leverage, or like I'm not saying that's more realistic than the seed idea, but it's more realistic than trying to learn these connections directly from cortex to hippocampus every time, because you basically have those object IDs kind of where you need them in the hippocampus. You can have these rapid temporary synapses and basically just reuse those IDs lots of times.
So I'm lost. Is this an alternate proposal to what I just presented? Is it different, or is it the same thing?
I think when we talked about it, it was, but thinking about it now, it's basically the same because the object ID would then invoke the unique reference frame versus what you just presented. It just kind of
I've said it
doesn't
right here.
show
This I said IDs here. So this was
yeah.
this is the same thing I talked about. I said the IDs would promote the right object reference frame. And I just drew this line here, but then I decided over here, I drew it at the top because that's where we put our object IDs. We show them coming off the top of the columns, you know. So I redrew it. Here's a little funny thing. I asked Vivian we had all these screen, you know, these whiteboard pictures, tons and tons of them. And I said, Vivian, can you share them with me? I couldn't find them. Tell me where they were. And I was looking for the one that was about this whole topic. And this is the only thing that was on there. It just like you said, I'm like, that's the only thing we wrote down from this whole discussion. Like, that's not much to go on.
We have this thing too. That one was, I think, the one that you just talked about, and then.
Oh, there was the one that was also included in it looks like it's loading, but that was included in our presentation. Right?
I'm not seeing it yet. It's still loading.
Yeah.
Anyway, it's all this as a humorous aside.
But yeah, I guess.
Question.
Could I? Well, yeah, go ahead. I'll have a further point.
I was just going to say that, yeah, for context to everyone else, I think this was maybe the last thing we talked about, like in the last few hours. So of the focus week. So yeah, partly it's just revisiting these ideas and thinking kind of what makes sense. Yeah, I think I agree with you, Vivian, that yeah, kind of functionally it's pretty much the same. It's almost be a question of like biological implementation.
Yeah, I think maybe this one on the right just spells out more explicitly that it's kind of like a voting connection to layer 2-3, and then it invokes the reference frame, like the classical mechanism we use all the time. Versus with the seed, the first time I heard it, I was thinking of it being a more explicit initialization in layer 6 directly, but
no,
thinking about it more, it should just be through the.
I didn't say how the seed would work. And maybe seed's not really the right idea. I mean, let's be explicit what the language is. The language is we have an object recognized over here. It has a unique, and there's an ID. That ID is sum total of all these neurons over here, right? You know, they all have the layer 3 neurons, but that's a unique SDR, and you can subsample from that and have a unique SDR. That unique SDR, which represents an ID, comes over here, and somehow, with mechanism unspecified, it tells how the grid cell modules need to be anchored. Now, there might be more to it than that, but at a minimum, it has to do that. And once you've anchored them uniquely to this SDR, then anytime I invoke this SDR, I'll have the same anchoring mechanism invoked here. And so I'll always invoke the same model. There's going to be more to it than that. This is just the first step of it, but it seemed necessary to do it that way. So now I can always invoke a temporary model of this object. So if this is the logo, I invoke a temporary model for the logo. And initially, it may have no features in it. It's just the right object. It's just the right reference frame. And so all we're learning is the new features that in this reference frame. I said this before or something again. Because
I mean, just for clarity, I guess the reference frame is special because it's unique, but because sometimes we talked about it kind of containing implicit information about, I don't know, the structure of the object, but I'm not sure it really would or needs to.
Yeah, I don't know. And there's a lot that can be wrong about this. There's a ton of things that are wrong about it. I just going to focus on, there's going to be a model here, and the model is unique to, there's a models here, and these models know what object they're on. And we need to invoke an equivalent column over here that says, I'm on the same model, even though I didn't have any data. You just somehow, based on what you know about the model ID, I'm able to create a blank model of the same thing. Does that make sense?
Yeah, I think that makes sense. And then, as long as you can, as you say, recall it from cortical input, or recall it from these hierarchical connections within the hippocampus.
All right.
Just
There is
recall the same reference frame.
there's an implication of this, but I didn't write down for today. But there's another implication. We have object IDs being shared here, right? And at any point in time, some columns are getting input and some columns aren't. And we talked about how other columns could learn if they're not getting input, like the shared learning thing. Well, it's possible that whatever mechanism that's able to invoke the correct model reference frame here, this is not an associative learning, right? I'm not taking a pattern here and a pattern here and associative learning it. I'm taking a pattern here and forcing a reference frame here. That same thing could be happening here. Maybe in the voting is not associative learning, but once somebody establishes something, maybe it automatically invokes the right reference frame over here. So just if this guy says, I know I'm on a mug and I'm sharing in my ID, then everybody else who's not getting an input or anybody could say, oh, I'm going to create a model of mugs too. I don't know that it's a mug. I'm just going to say, based on your ID, I'm going to create an equivalent model here. So it's like you could start with in some sense a blank reference frame, a blank model here too. Just like this could be a blank model. And of course, over time it gets filled in. But anyway, it's just the idea that the voting, we've always talked about the associative learning between two STRs or multiple STRs, and that works. But this implies that maybe it's not that. Maybe it's more like not associative learning, but an associative forcing function. Whenever we're not even associative. We all going to learn models, and we're going to somehow force the other models to be the same reference. I don't know. There's a lot to. Can I just go through my my questions before I don't want to make sure we forget them. Can I do that? And we can go back and talk some more.
Yeah, sounds good.
Yes. Right.
Yes.
So some of these I've already talked about. Is it reasonable to assume that there's only one column per grid scale? I think it's reasonable if I don't assume that those columns remember models permanently. If all they're doing is temporarily learning, compositional relationships, it's reasonable. Is it reasonable to assume that all columns have all models? This is a basic assumption of the thousand brains theory. And I've always wondered, like, is it all these columns in a region which all have to have all the models? Is it reasonable to assume that all can learn them? That's an ongoing thing. This one here. We just talked about morphological models. Is there equivalent relationships with behavior models? I have no idea. I didn't think about it at all. I just said, hey, we also have behavior models. Is there something like that going on here? I have no idea. We just talked about like compositional morphological structures. Are the cortical aware pathways related to the lateral entorhinal cortex? Remember there's two sides of the entorhinal cortex, and one was like objects, like what they are. That's what we've been talking about. There's the other side, which is sort of it was the episodic and where path. So anyway, in the cortex we have these where pathways, and in the hippocampal complex we have these lateral entorhinal cortex. And are they equivalent? What does it mean? I don't know. And then there's this concept of attention. We've talked about that a lot. And so I didn't mention that word at all in the above discussion, but underlying it, there's this idea that it's an hypothesis: when a novel unexpected input occurs, so the cortex says something's not right here, or something unexpected, the brain tries to infer the object. That is, okay, I look at the thing, it's a chip, it's the plate, whatever it is, or the car, the intersection. Infer the object where the novel input occurred, and that also invokes the episodic and fast learning in the hippocampal complex. So this is a subtle point. Observing an object in an area is not sufficient to invoke the hippocampal complex. It has to be unexpected. So just let's go back to this picture here. I've got something going on. I'm looking at something. There's an input which is unanticipated. I was already attending to some area to observe that object. I was looking at some space in the world. I said, oh, there's a car. It's unanticipated. Therefore, it's the fact that I'm attending to some area and it's unanticipated that invokes this mechanism over here. I don't want to invoke, I don't want to attention requires invoking this as well as some attended area. So it's not just the attended area, it's an attended area where something is unexpected that would invoke, you're saying, like, oh, well, let's pay attention to that. Let's remember this now. There was a car there. I invoke the episodic memory too, which is sort of an additional feature, like where was I and when did I do this? All right, those are my questions. And I'm sure there's many more. But okay, I'm done now, so I can stop talking completely.
I have a question. So when we have a temporary arrangement of some features, so these are happening in the hippocampus or the hippocampal complex. What happens if we have? Is there an equivalent of also a motor output from there? Because usually when you need like in an intersection or on a dinner set table, you can also, if there's a temporary arrangement of features, you can also reach to grab the plate or do something based on this temporary arrangement of features. So is it that there is a motor output from the HCC, or do we also have the opposite directions of the flow? Like these green lines also go the other way, and that would activate some motor output from the cortex.
Good questions. I have no idea the answer to that one. I can talk about one little surface related idea, which is just a slight variation of what we've already talked about, is that when I see the table, I walk into the room and I recognize the table, somehow I'm also invoking the temporary arrangement of features on the table. Like the table model includes not just what's the permanent table model, but it's the temporary. So these two things act together in a way, like the table now, the rice dish is on the far corner.
Mm.
It's sort of like together they represent the table. So there's the permanent features and the temporary features. That's all I can say about it. So when I think about behaviors, if you're saying, okay, I'm going to achieve some behaviors, I say, okay, well, on today's object, my goal is to put the fork or the rice plate in some position. And I say, well, I know where the rice plate is because my model of the table tells me where the rice plate is. And so when I act, I have to use the complete knowledge to act upon the table. But beyond that, I have no idea the answer to your question. Like, was there motor outputs here? I don't know.
Right.
But the way I want to approach it is to say, together, this is the table. It's the real stuff, the permanent stuff, and the temporary stuff. It's all one thing. I don't think of it as two separate things. It's just the table, and right now, that's where the rice is. And maybe there's a feature on the table that's permanent, never moves, and that's there too. So it's this, and then when I act on the table, I have to somehow act on both of these parts. So I don't know beyond that. It's a mystery to me.
The cortex would probably need some information from the temporary arrangement to be able to.
Right. That's what I'm saying. It's like the temporary arrangement is part of the model now. It's like there's not like it's two models. It's like it's one model of the table, including the permanent features and the temporary features, and you can act on that as if it's one thing.
And those directions,
Yeah.
the green arrow is probably bidirectional, or at least there's a similar type of connection going back the other way.
I don't know how it works.
Yeah.
I have zero idea.
That's actually what got us to this topic in the first place. And then we didn't have enough time to come back from, okay, how can we learn temporary arrangements back to, okay, now we know the temporary arrangement. Now, how do we modify it to the goal state? So that would have probably been the next discussion topic that, we can also talk about.
Continue on for another day or something like that. But excuse me, Hoji, but many of these topics I just brought up was things I was bringing up in the past, independent of that particular problem. I was just not thinking about attention and so on. But yeah, you're right in that we did talk about this idea of acting on things. You need to know where the temporary position of things are to act. Sorry, Hoji.
Besides, I think this is somewhat related to temporary models. Besides maybe accumulating different features like a chip in a mug, I think there's also the role of context in perhaps in forking, maybe. So the example that I'm thinking of is let's say you have two identical bedside tables, one on the left and the one on the right. So they're identical in terms of features and morphology, But at some point, it becomes like one becomes Jeff's bedside table and one becomes Janet's bedside table just because of the context at like the location it is, the where it is placed in the bed, like relative to the bed, and somehow like now that table, because of that context, even though there's no change in features and morphology, eventually we might have like two separate object IDs, one for Jeff's bedside table and one for Jen's bedside table.
Right.
Maybe.
That seems like a practical problem. This bedside table problem, which makes it interesting. I think if we spend some time thinking, we might come up with a solution. What we don't want to do is ever have to. We really want to avoid to fork a model, having to relearn the model. Right. So think the two bedside tables. Maybe there's a feature of since I have two different IDs for those bedside tables. I don't know. I know them separately, right?
Yeah.
Because of their context. And now I discover there's some new little feature inside the draw. I would immediately assume it's existing on the other one too. It's like, oh yeah, that's part of this table. Therefore, I never noticed it before, but I've seen it on this one now that it's going to be in the other one. So that tells me that, somehow I'm using the same model. I'm updating a model of the generic table while at the same time knowing there's two versions of it.
Yeah, that's kind of an interesting example because it kind of depends on what the feature is, I feel.
Right.
Well,
And also, like,
I was picking a feature that is clearly part of the table design.
Right.
Like there was
somehow you're able to use.
Imagine if you don't know how to remove a table sometimes, there's often they're on slider, a draw. It says a draw. To remove a draw, there's often a little mechanism underneath the draw in the slider where you have to pinch and so you might have discovered that. And as soon as you discover that, you're going to assume it's on the other table too, right? So there's an example. If I saw an ink stain in the draw, I would assume it's not in the other table. I would assume that's unique to this table. I don't know what to make of that, but your point.
Maybe, I guess compositionality maybe helps with there as well. Like you have a model of the slider and stuff, but that's an interesting one.
Well, it does. The composition really does that. If I say there's a room and there's two identical features in the room, that's no problem. Our models can handle that. No problem, right? We know that there should be this feature here. There should be a version of this table here. There should be a version of this table here. But now somehow we have to say, in the context of the room, I have to remember child features of the table that are different. And that we haven't really discussed before. If I said the tissue box on one side of the bed was in the context of the room, I'd have no problem. It's only when I put it in the context of the table, then I have a problem because I only have one model of the table. So it's almost like I have to have three sort of compositional levels or something like that. This is a problem that can be solved pretty quickly, I think. The multiple tables problem.
I think the example that Dale sent in the chat is a pretty interesting one to think through as well, which is the gym lockers, where you have a bunch of lockers that look exactly the same. And then depending on which locker, where it is on the wall, there will be different things in them because different people own them. And it seems like we can learn very quickly different instances of lockers. Like when we went to the Twist Museum and we shared several lockers, we would know, depending on which locker we went to, whose stuff was in there. But we didn't like see it repeatedly or learn different instances of the different lockers or think, oh, this locker belongs to Niels and Scott, or like it was pretty much instantaneously just based on where the locker was.
Actually, can you remember where your locker was now?
Yeah, I remember.
Only by attending to it. If you don't attend to it, that's a great way to lose your stuff.
Well, but I'm saying right now, 2 weeks later, I was just curious if your episodic memory.
Yeah, I remember Niels and I had stuff on the left side, and then others had
top left,
more in the top right corner.
Right.
Yeah.
So I just testing how long
mine was on top right.
I'm assuming you're going to forget this. Remember HM remembered everything up to about a month before her surgery, which implies it takes about a month before you can reliably get things in the cortex. So we should ask this again in like 2 weeks. But we just
now.
So
I was gonna say now I'll definitely remember.
Put a 5 weeks from now. Put a little thing and say, what locker do we use?
I'd like to pitch a thinking about solution framing that maybe it's useful, maybe helps with two problems, which is, like you said, there's two tables and you see something on one, you immediately find it on the other, but like an ink stain would not translate. To me, it's the analogous of you're in two identical rooms and you locate yourself in one and you know you're not in the other. And then when you walk to the other room, you know you're in that room and you're not in the other. It seems like, us being part of the compositional object for the purpose locating yourself in space is the same problem. But maybe just thinking about it from that framing also might also bring about like how do we actually solve that? Because
Well, that's
it's
interesting.
right.
Right. Well, I was just translating it to the classic example of a rat in a maze. This is where grid cells come about, right? The rat recognizes the room and it keeps track of where it's in the room. So let's assume you already know where your body is relative to the room. That is the context. I think what you just said, Tristan, is that is the context you could use to differentiate the two tables. It's not anything about the tables themselves, perhaps, but you're saying maybe where I am in the room.
What I'm saying is I'm making the analogy between a room and a table and ink stain and the entity. I can tell the two tables apart because one has an ink stain on it. I can tell which room I'm in because I am in one. That's the analogy I'm making.
Right, okay. I was assuming the ink stain, by the way, was hidden in the drawer. So the basic assumption is you're looking at the two tables, they look identical, except one has a tissue box on it, one doesn't, or something like that. But I think your analogy is right. It would make sense. I'm not sure I understand it completely, but
or another analogy would be I'm making an analogy between a cup and a room and the chip and the entity in the room.
Right. Well, that's basically, that was the original derivation of the thousand brains theory. We had, I don't know if you remember this, but the very first papers on the thousand brains theory, they showed a rat in a room and a finger on a coffee cup, and it said these are equivalent. Your finger is like the person, and the coffee cup is like the room. So if that's what you're saying,
but it doesn't sound exactly the same, because if you're making the analogy to the chip on the chip on the mug doesn't move. Like you can't go to a different location on the mug.
Right, but that's equivalent to the table in the room, right? The table in the room is equivalent to the chip on the cup.
Yeah.
Are we talking about what you're talking about, Tristan? Or are we lost?
I was addressing that we have two, like let's say the stain on the table is not hidden, it's just there. And it's like, and you can tell apart that this is the table with the stain and this is the table without a stain. And we're talking about all the mechanisms that work between the regions and the hippocampus, etc. And what I'm saying is that's one way to frame the problem. And another way to frame the problem is how we do navigation is where we know where we are in the world. That if I am a stain in the room and the other room doesn't have a stain, doesn't have me, the other room doesn't have me in it, I can tell the tables apart, I can tell the rooms apart because I'm in the room that's with me in it.
But it still seems different because you could just go into the other room. You're not a feature of the room.
Right. So imagine, okay, so bad example. There is a cup on a table and there's a table without a cup on it. I can move the cup onto the other table and identify the table by which one has the cup on it. Like that's the distinct from the other one.
Yeah.
And I can track which one it is.
But but in general, those tables are identical. It's just right now it's more, I would say it's the other way around. I would say, if I know what table I'm on, I'll know if it has a mug on it or not. Like I could see a mug not on a table at all, and I wouldn't say, "Oh, where's the table?" I'd say that's just a mug. It's in the cabinet.
Yeah. Okay. All I'm trying to do is show a mapping between framing of compositional object identification and navigation in the world, and then maybe we get some insight from thinking about navigating in the world that will help us solve compositional objects. That's what I'm trying to do.
Yeah, right. Well, I think Yeah, I mean, ultimately have to bring that part of the picture in, right? Where you are in the world and where sensors are, and where's your body. Okay, it's a good point.
I think in order to make that analogy work, you have to do something like you're in a dorm where all the dorm rooms they are the same, except for, like in the way that all the tables might be the same,
Yeah.
minus some. You have to be in a place where all the rooms are basically the same, but can be identifiable by some feature. Which,
Yeah, like generally, I feel in that case, you recognize what room you're in by how you got there. So in a higher level reference frame. And if you didn't have access to that and the rooms were literally the same, you wouldn't know which room you're in.
Right.
yeah.
If you were just teleported into it.
If you're in an infinite hallway of doors, you couldn't tell where you are.
Yeah.
Right. So,
And
But it's
so
still another example of the model forking. If you're in a hotel and you know one room and then you go into the next room, you don't have to learn the whole layout. You can predict there'll be the same layout,
right.
except for maybe your suitcase being in the other room. And yeah, you can fork.
I think I'll make a statement now. I don't think models fork. That is not in the way we think about as computer scientists. So, we don't copy a model. There's no copying in the brain. So somehow all these things have to be done with sort of some level of compositional structure, meaning like, okay, there's the model of the room, but I now know the additional context of it's the end of the hallway. And so I can add features to that model of the room that are only in the end of the hallway, room versus another one. I'm just speaking out loud here. This would be a good topic for to think about for the next week or two. Really address the quote forking problem. How is it that I have let's take the mug of the chip. All right, I have the Thousand Brains model cup, Thousand Brains cup model in my head. I have a bunch of those. I bought a whole bunch. I have a cabinet full of them. Well, not full of them, but we use them. And now I have one that has a chip. Okay. How is it that I represent the one with the chip? I don't believe I have a new. I'm sure now that I don't have a completely new model. It has to be a model with the extra chip. And yet we've just talked about how to do that temporarily with the hippocampus. Like I see the chip for the first time, I can say, oh, I now have a separate place where I'm storing the chip part versus the rest of the mug part. Going back to that. Am I still sharing?
Yeah.
I think I'm still sharing.
Yeah.
I'm sorry. You guys see? Hang on a sec. So we go back to this idea here is that we're just storing the chip variation here, but all the mug is the same here. And my point is, I don't think we fork models here. Somehow we have to do it with either hierarchical composition or something like this with the hippocampus. The variations of a base model have to be they somehow have to be done they have to be done with linking, not with copying models. I'll just state that. So this is an open question. How do we represent these variations, whether it's variations of end tables, variations of mugs?
Is the crumpled t-shirt also a variation that we represent in the hippocampus?
Should I stop sharing? Maybe I should stop sharing. Say it again, Rami.
Is a crumpled t-shirt also another variation that we represent there, or is it different?
Well, we talked about how to do that with the lower dimensional models. That could be part of the solution.
Yeah, like in terms of the higher dimensional model, like if you're really focusing on it and attending to the t-shirt, learning kind of like a temporary 3D model of it, probably would.
Yeah, it's a temporary variation of the features.
I don't know. It feels different to me. That seems like a different problem, but
I think we know yet. You would learn a temporary arrangement of the t-shirt, but it might not be invoked by the t-shirt ID. Like you might not use the t-shirt model as a baseline for adding extra features. It might just be a new temporary morphology model.
Yeah. Yeah. In general, I feel like the whole invoking thing and the importance of that is, or like how that mechanism works is maybe one of the bigger.
I feel like it would be more like we're invoking. We still use the object ID of the T-shirt, and then we seed and we create basically a reference frame, and then it's just different arrangement of features, like the collar and the like different
Well,
parts of the T-shirt on there, and it's just arranged differently.
I mean, I feel like the 3D model of the t-shirt, like the canonical shape, can't really tell you anything about the crumpled shape. So it doesn't help much to seed a reference frame with that or associated.
Yeah, but
So you can
it
just,
is helpful to associate the semantic ID.
I mean, yeah, you can recognize the 2D model of it and use that in both cases. But if you want to learn a temporary arrangement of the 3D morphological features, it doesn't seem like the 3D morphology of the canonical t-shirt would help much.
I'm not following all these words, but I think we're saying, let's see if I got this right. Um, the deformation of a t-shirt to me seems fundamentally different than the, crack on the coffee cup, uh, or the temporary arrangement of features. Um, is that what we're saying, or are we saying the opposite? To me, those seem like very different problems.
Yeah.
Yeah.
That's different than the intersection problem because it feels like it's just a more complicated, like more complex. It's just a lot more features that are crumpled or.
Well,
Well, I
it's
mean, at the intersection, you still have the base model of the intersection, like where the traffic light is and where the sidewalk ends and all that. And those are still useful to make predictions. You're only storing the temporary stuff like the cars.
when you said the intersection, I thought you meant like the Venn diagram intersection. We're talking about the intersection of these two
Oh,
problems. You're actually you were talking about the
The
physical
traffic,
intersection. I'm like, what the hell are you talking about? The intersection? What intersection are we talking about?
not the
Sorry.
conceptual
I
intersection,
was I
the
was going
physical.
down this I was going down this idea, imagining intersection of two problems, and I'm like, God, how's that work? And I had no idea what you were talking about. Okay, and then you said cars. Like, oh shit. Sorry. Keep
You
going.
anchored the wrong reference frame.
I anchored the wrong reference frame, right? I anchored the Venn diagram reference frame. That's my canonical image of it. The two circles, the thing in the middle. That's my canonical image of a reference of the intersection versus the one with the car.
Yeah.
Sorry, I have no idea what you were talking about now. Go
Well,
on.
I'm just saying that with the car intersection, it's still features that are just in different temporary orders.
It's not even that. It's new features too, right? It's not even just the same cars. It could be anything. It could be somebody could be on a moped. Somebody could be flying a kite. It's just like, here's some structure and there's new things on it. And yeah, they are some of them are moving, but it's not just I'm deforming the intersection. It's a whole bunch of new features that it didn't exist before, and now this is their temporary arrangement. With a
No, but
t-shirt
if you were
example,
going to learn, like, if you were going to actually attend to and focus on learning, like the t-shirt, that would, like the crumpled t-shirt, that presumably would happen in the hippocampus. I think the distinction you're making,
right?
Vivian, which I agree with, is that it wouldn't leverage existing models in Cortex in the same way that we could with like the chipped mug, because like the dimensionality that we're trying to learn we don't have an existing model that's useful for that. So to me, it seems related, but it's not the exact same as the case of like the mug with a chip.
And with the intersection, it is helpful to leverage an existing model.
Right.
You have the model of the streets and the traffic lights, which are constant, and then you're adding new features into it, like the cars, in your temporary model.
Once again, for years we've been bouncing around these issues of different classes of objects and deformations of objects and scale changes of objects, and now we're talking about temporary arrangements of features on objects. These all seem to be they're all related in some way. They're like, and it feels like an answer is going to pop out. I'm sure hopefully most of these things will get resolved at once. I think the thing I presented today and what we talked about in London is another ingredient to help us think about that solution. It's not the solution.
Another connection I just made when you said this thing earlier about how voting could be a stronger thing, not just a way to narrow down existing hypotheses, but to basically enforce a hypothesis onto a bunch of columns.
Right.
That relates back to what we talked about with shared learning on like the first or second day.
Right.
You were saying
Right.
all columns in a region are recognizing the same or representing the same object, even if they're not getting sensory input. And that mechanism could actually be the way to implement that.
Right. I thought I was saying that, but maybe I didn't get across. But
Well, yeah, you were. I just didn't realize the connection to the shared learning topic.
Okay.
So
here's another thing. Remember the abstract I showed you from that paper? Just a couple sentences. I didn't read the paper, but they were talking about place cells, right? And the place cells were like invoked at the same time, like cells that in V1 that fired at a particular location. They would have an equivalent cell that was firing at a particular location in the hippocampus. That's more than I don't. That wasn't. It wasn't. That's not what I said. I was talking about invoking reference frames. And so I'm wondering, talking about shared learning, is it somehow possible that you're specifying the reference frame and the attended feature at that time? And so that would really solve the shared learning problem. It says, okay, we're at this location. I'm at this location. Here's the feed. Well, I don't know. That's not going to work. Shit. All right. Anyway,
if the shared feature is an object ID,
yeah, then it's fine.
I guess it
it's
would work.
Yeah, but then but I can't share individual features of columns or learning modules.
Vivian, your idea sounds like you could learn morphology without replay, right? Because you would get location movements even if you're not sensing any features or whatever, because the reference frame is forced on you. Then as long as you don't jump to a different reference frame, you can probably assume that where you are is the actual point on the object in the column that's not receiving any features.
You mean as in like from the subset of movements that we get? Like, I think that's what Vivian was describing last week with like, like even though we're not getting sensory input, we're kind of assuming that like this is a significant point. So this is a point in the model. Is that what you mean? Or?
Yeah, but you don't need replay anymore.
Why not?
Because the voting tells you what reference frame you're in, so you don't have to replay it. Every movement is that reference frame at the moment it happens.
It tells you the reference frame, but it doesn't tell you where in the reference frame you are.
Right.
I just want to point out, Tristan, you've been forked. There's two versions of you on my screen. I don't know what that is.
Which one am I? Which one am I?
Well, the one on the right's talking. The one on the left is muted. Now the one on the right's muted, and the one on the left's muted. So you tell me.
It's just
the same picture too.
It's just a really elaborate plot to get my point across.
Okay. What if you turned on your camera? What would we see? Are you going to turn on your camera? So the one on the left, I got a picture of you, and the one on the right, I have a different picture of you. You have two cameras. Okay, I don't know what you're doing. What the hell? You're logged in twice. All right, sorry that we got off topic there.
I think maybe a longer follow-up discussion could be like what we can expect seeding to achieve in terms of like information that can invoke whatever, because like it's not going to like give you literally a model, if it's essentially just saying this is a unique reference frame, then we're not asking it to do much, but obviously it's more limited in what it can do.
Well, it's asking to do something we haven't thought about before. And if I think about the neurons, I don't know how the hell they're going to do that. But but it's from a theory point of view, it's not asking too much. From a neuron point of view, I'm like, what the hell? I don't know what those are. But it's funny when we talked about the hippocampal building temporary models. I didn't need any more than that. I think the whole thing works just with object IDs. Right? I didn't need, because I have two levels in the hierarchy representing two objects, and then I have two models of the hippocampal complex, and it works. I didn't need any additional information. But now we've come up with other examples. It seems like we want to share, like in the shared learning, we want to share more than just the object ID. And that's where the replay came in and all that stuff.
Yeah, I think maybe one visual way one could think about the reference frame initialization is if you think about the universe and each object is a star in the universe. At each star, one object is being learned. So that they are like really far away from each other. There's no object that would be large enough that it would overlap with another object. So when you get the seed, it tells you which star you're at, so what location in the universe. But then there you still have to get relative displacements to learn the object and the features on the object, if that makes sense.
Right. Yeah. Well, I think your star analogy just describes the nature of SDRs, right? Each one's a point in a very, very, very large space, and they're by definition they're far apart. And even if you get noise in your SDR, you're still unique. But it doesn't explain how I get my reference frame, because I want a unique reference frame for that star. I'm just saying, just knowing where in the universe you are, like which star, it doesn't tell you the object that, like the shape of the object that's there, or any of the features that are there, unless you have learned them already. Right. I'm going to say it again, just to repeat, because sometimes it's helpful to do that. The mechanism we just talked about, where the hippocampus is learning, it's essentially saying I'm taking a model of something and I'm dividing it into two parts. And somehow those two parts have to be, they kind of learn separately, but they can be acted upon as one. Again, like going back to the question of I'm going to move the plate. I have to know where the plate is on the table right now. And somehow this is like this idea that you have a model that's divided into parts, yet they act as one, is an intriguing concept that somehow might relate to all these problems. I don't know. I can't say any better than that. It's just like a visual imagery
Is that
in my head.
The 1 minute summary?
Do you want me to do a 1 minute summary? Are we ready for that? Are we wasted time to call it a day? I'm happy to do that. Is that what you want right now?
Sure. I could also do one for the ultrasound.
You probably should have done that earlier. But you want to do the ultrasound first?
Maybe I do that after you, and then that can be a separate recording.
All right.
I also prepared some notes on the other topic we discussed during last week, but I don't know if we have enough time for more.
I have enough time. I didn't have enough mental bandwidth to discuss the current topic longer, but I think a new topic would work for me. So I don't know about anyone else.
About the motor commands and motor outputs and target pose and goal states.
I would like to hear that. If other people can't, I would like to hear that.
Sure. Yeah.
We can still summarize this part if that's easier when it's fresh in mind.
Can we take a quick bio break before we?
Sure. Right. I'll fill my coffee cup. Back.
Are you taking a break, Vivian?
I'm still here. You have a question or thought?
Sorry. So let me turn on the right things. So you know how we talked about location being from thalamus and layer 6 versus features coming in elsewhere. So what I was trying to say is, if you imagine that the column region, and if they're all getting forced into the same reference frame, I don't know if this is true or not, but each column is still getting thalamic input as it's moving around in the world. And so it's not getting L3 features in, but it's even though if it's not getting any L3 features, it would still get displacement in L6. And as long as that voting forcing mechanism isn't shoving it into a different reference frame, then maybe that's what's letting it learn morphology in each, even though it doesn't receive any features. That's what I was trying to get at. Does that make sense?
Yeah, but that assumes that they are still all getting displacements as input, right?
Yes.
So that's basically like the replay mechanism.
But what I'm saying, you don't need a replay. You can do it live because the voting is forcing you, already telling you what reference frame you're in.
So you're saying at every step, even though only
Yes.
one sensor is moving, you're sending the same movement to all of the sensors? Or all of the columns in a region?
Yeah, I see what you're saying. Okay, thank you. I understand where there's a gap. So that's what I want to clarify.
I didn't even. So what you realized now was it doesn't work with like moving your hand, but it could work partially with moving your eyes, or.
Well, if I'm moving my hand, that moves all the sensors.
Yeah.
And so but my left hand is not learning what I'm touching.
Yeah.
Because it's not getting any position movement. So this proves what I was trying to say. So thank you for highlighting that. Appreciate it.
Okay, cool. I'm not 100% sure how I highlighted it, but
no, I get it. I understand the need for replay in that case. I was coupling all my sensors together when they're decoupled. That's what the problem was.
Yeah.
I still don't like the replay idea. I mean, it kind of sort of worked. It just seems kind of janky.
Why do you think?
Well, first of all, it's very limited how much replay you can do, right? And I don't know. It just feels like really kind of asking a lot. I mean, I know we see replay, so it's like it exists, but it just seems like to train all these columns, all these different places. I don't know. It just seems a bit hard. I'm not sure. It's very limited. Like memory can only do a few points at a time in the replay. And so not everybody, then we have to decide which columns are going to be replaying and, who gets to decide what's being replayed. I don't know. Well, replay always occurs. It's been observed in the hippocampus. If that's a source of, I forgot. Are you going to talk about that now? Is that what you're going to talk about? What are you going to talk about now? No, something else.
No.
I was getting a clarification from Vivian. So
I know I'm just riffing here. I'm just thinking of other things. I was just talking about how I don't like to replay for shared learning, and one of the reasons is it seems like all these columns, a bunch of columns of learning, and then who gets to decide what's being replayed, and because each column is different in some sense. But I'm thinking but replay was associated with the hippocampus, so there was one episodic trace in the hippocampus, and if that was replayed for everybody, which is a standard dogma, that's the standard dogma for this. Would it work? I don't know if we talked about the hippocampus playing a role in the replay when we talked about replay for learning. Did we?
I thought that was assumed because that's where it comes from.
Yeah. Well, we didn't say a specific place where this type of replay would happen, but just that it's observed in the hippocampus.
But we did talk about there's multiple columns that could be replaying, or at least I was confused by it. I was thinking who's going to dictate who's replaying? But if it's a hippocampus, there's only one trace.
Yeah, that's the way
we didn't say it would be the columns. It would be some shared space that
like the hippocampus that has one representation
that
quickly develops and then replays.
I don't think we at least I don't remember walking through thinking about how the hippocampus would do this. In the issues of, because now we're talking about this information going from the hippocampus back to the cortex, which I didn't have that error in my picture. And Okay, so that's next time we talk about replay, we have to really think, what if it all is derived from the hippocampus? What, given our new thinking about the hippocampus, what does that tell us? Okay, we're going to leave that aside. Just try to remember to talk about that next time.